His topic is point mutation assays and resistance testing in HIV management. Dr. Kantor is Associate Professor of Medicine at Brown University in the USA. His main research interests are HIV drug resistance in non-beach subtypes, the development of sensitive point mutation assays, and their potential use in India. So now I'll invite Dr. Rami Kender.
Thank you very much. It's a pleasure to be here, and I thank the organizers very much for the invitation to talk. I'm going to speak about drug resistance, and we're going to hear more of an overview tomorrow. And so I'm going to talk just a little bit about drug resistance and drug resistance testing to give you an orientation to what I'm going to talk about. The majority of the talk will be on point mutation assays. You've heard them mentioned before by Dr. Katzenstein. I'll get into some more detail about what they are and why we want to even consider them in the context of drug resistance testing in India and beyond. And I'll talk a little bit about the current experience that we've had and other people have had using the various assays. And I'll try to concentrate in India as well as in global settings. So drug resistance is important. I could provide a lot of data for this, but what I'm showing here is a report from the WHO from this year talking about their plan for the next 5 years to roll out drug resistance surveillance globally. And the main reason that it's important is because treatment access is increasing, like everyone has already mentioned, and when more people get treatment, more people will develop drug resistance, which will then be transmitted to other people and which will impact failure. So this circle is going to happen and is going to increase, and it's going to harm, like Dr. Katzenstein mentioned, the last portion of the 90%. So drug resistance is very, very important globally and will continue to be. One slide on the mechanism of drug resistance. People are familiar with this, but we're talking about drug resistance mutations that occur in proteins that the drugs act upon. On the upper left is the reverse transcriptase, and the drug is here, and the active site of the enzyme is around here, and those red, green, and gray mutations are those that develop as a consequence of that in order to make the drug not be active. And the same thing is on the upper right in the protease and on the bottom right in the integrase. So we're talking about specific mutations that develop in important areas of proteins that matter and do not let the drug do what they're supposed to do. So the conventional way we test for these mutations, and this was already mentioned as well, is what we call conventional or Sanger sequencing or population sequencing. Those are the terms that are used. We're taking a blood sample from our patients. We're extracting the RNA of the virus from it. We're reverse transcribing it to DNA, amplifying the DNA, sequencing the DNA, then translating from nucleic acid to amino acid, and we come up with this list of mutations that we then say this is what our patient has and this is what the treatment regimen should be that would be susceptible based on those mutations. And so this Sanger sequencing is a relatively old method that's been used for more than 40 years. It's expensive, it's labor intensive, and it's time consuming. We're still using it because this is the validated assay, and that's the most common assay that's used around the world. And it requires laboratory infrastructure in order to do it. It provides us a full sequence. So we get a list of all the mutations that exist in our patient that are shown by just schematically by all the arrows. So we get the full virus, and we can identify specific mutations that exist throughout the virus. But there might be a sensitivity problem. So if you look at this graph, the population sequencing or the bulk sequencing that we can detect with this method, the Sanger sequencing, is only above 20% of the overall virus population in the body of the patient. There is a lower limit where we can't detect it with this mutation, and this lower limit or low frequency or minority variant resistant variants that we're talking about can be significant. So the Sanger sequencing is less sensitive than what we might need to know, and resistant variants might exist in minority variants, and so when we do Sanger sequencing, we might underestimate the resistance. So there's two things. In Sanger sequencing, we're getting the full virus, and we might be underestimating resistance because of sensitivity. And this is where point mutation assays come in. Point mutation assay review, I'm citing it here on the bottom left, and this is a table from that paper, and so anyone who's interested more can read that review. But there's several options for drug resistance testing that are shown in this table. We'll talk a little bit about allele-specific PCR and about oligoligation assay, but there are other methods of drug resistance. But what it does is it increases that sensitivity that we discussed before. So if you look at the graph, in the gray, it's population or Sanger sequencing, and in black, it's the point mutation assay. Doesn't matter which one you choose, and you can see that the black is higher than the gray in general, and each one in the x-axis is just different mutations. So the bottom line of this is that we can detect more of those drug resistance mutations if we're using a more sensitive assay. And the threshold that we're detecting it by matters. What this second graph here shows is that if you go lower with that threshold, it's not necessarily good. In the lower left, I can't use the mouse, but in the lower left bar right here, the difference between the threshold makes a larger difference to treatment outcome, whereas if we go lower with the threshold, that difference decreases. So the bottom line is we need to know what the threshold of this assay is and what is its accuracy. So the fact that it's more sensitive might not always be accurate. So Dr. Katzenstein mentioned this, but when we're talking about point mutation assays or single mutations, you'll tell me, well, the Sanger sequencing provides a full sequence and all those mutations. The point mutation assays provide single sequence, single mutation, so is that good or bad? And the paper that Dr. Katzenstein mentioned basically addressed that. And it's from the Stanford database, and they took thousands and tens of thousands of sequences and basically asked what are the most common mutations that we might want to look at if we had this point mutation assay. And so what they did is they looked at tens of thousands of sequences. You can see them here either in lower income countries or upper income countries. And they looked at the cumulative incidence of those mutations and try to come up with a list of what would be the most sensitive and specific list of mutations. And you saw this graph from Dr. Katzenstein basically said these 6 mutations that appear here on the left, if we had a point mutation assay, let's develop it on these mutations. And this is what's guiding the point mutation assay field. So let's review a few point mutation assays. So PANDA is probably the most successful one, the most advanced one, and you'll hear about it tomorrow from the chair, so I'm not going to talk about this. I'll talk about a couple of other point mutation assays. One is oligonucleotide ligation assay developed by Dr. Frankel, and this slide is from her. She's from the University of Washington. And again, without getting into a lot of details, what this basically does is it amplifies the virus, and then it does ligation in order to find if a mutation is there or not. If a mutation is there, then you get a different color in the virus of our patient. For example, in this case, yellow. If a mutation is not there, the ligation would not happen, and you'd get a blue. And so you'd be able to say mutation X is there or not there based on this assay. And this is what point mutation assay does. So what are the strengths of point mutation assays? This is for OLA, but it's true for other point mutation assays as well. It's sensitive. We talked about that. It could detect lower levels than what Sanger sequencing does. It's specific. It can quantify the minority variant frequencies. It can tell us this mutation exists in 16% or 2% or even 98%, something we cannot do with Sanger sequencing. And it's relatively simple. You need minimal equipment and not this whole huge lab that we discussed originally for Sanger sequencing. What are some of its weaknesses? It needs technical skills. It needs development of the assay. You sometimes need to batch specimens in order to get to the low cost that you need. Turnaround time might be not that short as in a point of care concept that we heard before that might be important, and so this might not be a good point of care method. These assays, once they've been developed, need to be validated, and OLA was validated. This is one example. The graphs show a great concordance between OLA and another method that we'll talk about tomorrow, which is next generation sequencing, and it basically shows that there's a high concordance in the detection of the specific mutations with OLA, which is wonderful and is what we need. Dr. Frankel and her group also showed that if we detect those mutations using this point mutation assay, we can predict treatment failure. This was a study from Kenya in 236 patients that were naive to treatment, and they were looking if those patients that are naive to treatment had drug resistance mutations, and they found it in 3.9%, and they found that those that had this resistance tended to fail more, even tenfold higher, and you can see it on the graph on the right, the difference between those with and without transmitted drug resistance. And so if we have this assay here or anywhere else, we can prevent suboptimal treatment on a very sensitive way. So a couple of other point mutation assays that are in different levels of development. This is one assay that we developed about 2 or 3 years ago. We called it SMART, Simple Method to Amplify RNA Targets. Without getting into too much detail, what we did here is we were able to isolate, we were able to isolate virus from the patient, then use magnetic beads that have specific probes on them that can detect the mutations, and then use a magnet to, on a chip that's shown here on the left, move those beads with those viruses to a different well into which we can then amplify the RNA and then detect it similar to OLA. Again, the details are not very important, but it's just another way of trying to do it in a different way to get to the detection of specific mutations. And the same thing is in this method, which we developed relatively recently, and the idea is the same. It does ligation, but it does it on RNA as opposed to on DNA, which has several advantages. Bottom line is there are additional methods that are being developed and might have a place of importance. The last method I'll mention is the allele-specific PCR, which is not a new method. It was developed originally in 2006 by a group in South Africa. And the main bottom line of this method is shown here at the bottom, whereas basically we designed primers that would identify the mutation of interest by looking at the differences in the nucleotide or the sequence shown here in the different arrows, and the red arrows that show the different options that can be detected. So if the blue plus the upper red would be what the patient has, then we'd be able to detect this one, and so on and so forth. Basically the same type of idea of, that we've been discussing up to now. Without getting into too many details, the bottom line here is that whenever there's a mutation, it will amplify. Whenever there isn't a mutation, it won't amplify. The same type of concept we discussed. And we apply this, developed this actually at YRG Care in Chennai, based on subtype C that's specific for India. We did it, we presented it in the AIDS conference this year, and we did it in 83 adults. We did it both in RNA and in DNA, and we used both population sequencing and allele-specific PCR using a different mutation than what I showed you up to now, which was on K103M, but we did it on a K65R, which is a tenofovir-based mutation. And what I'm showing you here is this is the drug-naive people that we did this allele-specific PCR on, and you see that in RNA there was no K65R detected in the RNA and no K65R detected in the DNA. But when we did our allele-specific PCR in these 4 different thresholds, either 2% or 1% or 2% or 5% or 10%, you could see that the frequency of K65R was detected as opposed to the 0 with a Sanger, but it was detected in decreasing frequencies based on your threshold. And then the same idea was in the DNA, exactly the same frequencies. And if we look at the people that were treated, in the Sanger, we did detect those K65Rs. You can see the percentages here either in the RNA or in the DNA. When we did the allele-specific PCR, you see the same type of picture that we saw on the left, only in higher frequencies, and the thresholds really matter, which basically means this needs validation, which we're currently doing in order to say what is the most important or most significant thresholds that we need to use in order to implement this in the clinic as the next step. Allele-specific PCR has been validated in the US, not based on India samples, and so I won't go into the detail, but basically the bottom line of this is that it shows a lot more mutations that were identified in this validated assay compared to the Sanger sequencing. So this is my last slide before the summary. New directions, I showed you the picture of the chip. This OLA was developed based on a strip-like paper, and this is a future direction of the OLA group, and that might be a point-of-care test that can be used in the clinic because a paper will be able to tell us which mutation exists or not. So to conclude, transmitted and acquired resistance is a major problem for patient care in India and globally. Point mutation assays detect valuable and maybe hidden resistance and should continue to be developed. Global incorporation of drug resistance testing into clinical care, I think, will eventually be inevitable, and the role of point mutation assays needs to be determined in that. So thank you very much.